This video will cover subtopic 2.1 of the molecular biology unit. Here are the list of understandings that will be covered in this video, as well as application and skills. So we'll start with just a general definition of molecular biology. So molecular biology is considered a branch of biology that is focused on the structures and functions at the molecular level. And this whole molecular biology is a rather large division of biology itself. So molecular biology will look to understand, for example, the processes of cells and how these processes occur. Things like cell division, energy creation, energy utilization, cell specialization, and cell communication. And we'll look to understand how these processes happen through the various substances involved, which include things like water, inorganic ions, such as sodium ions or chloride ions, for example, polysaccharides, things like starch or cellulose, or even carbohydrates themselves, such as glucose. It can involve proteins, fatty acids, vitamins, nucleic acids, which for us are DNA and RNA, and how these substances contribute to these processes, or how they are involved with these various processes that a cell carries out. So when we consider all of these various compounds, like they are considered organic compounds coming from living things. And so these organic compounds are obviously produced by these living organisms, but can also be artificially synthesized as well. So the example for you to know is that of urea. And so urea is a molecule and a main component of urine. And urea is created within an organism in a rather complex cycle that will ultimately produce urea as a byproduct. But it turns out that urea can also be artificially synthesized in the following reaction. So if you take ammonia with carbon dioxide, you'll get an intermediate product, ammonium carbonate, which will decompose into urea and water. So you can get the same molecule created by a different reaction, and it doesn't have to occur within a living organism. Now, for a long time, it was thought that only living organisms could produce these organic compounds through a theory referred to as vitalism. So this is a theory that the phenomena of life is due to a vital principle. And this vital principle was different from chemical or physical forces. It could not be explained by these chemical or physical forces. And this vital principle was thought to be required for organic compounds to be produced. So basically the thought was that these organic compounds could not be artificially synthesized. They could only be created in living systems. However, in 1828, an individual by the name of Frederick Wohler synthesized urea using silver isocyanate and ammonium chloride. Now, the significance of this was that this was the first organic compound to be artificially synthesized. And Wohler did this with no vital principle being required. It was very basic compounds that he was using, and he created urea. And so many compounds from living organisms have been created since, and has shown that this vitalism, or this theory of vitalism, is not something that holds true or stands pat. So all of these organic compounds have carbon as its central atom. And carbon is very important forming these organic compounds. So we'll take a step back and look at carbon and what makes it so useful in creating all these compounds. So carbon has in its nucleus 6 protons and 6 neutrons. So any atom that you find anywhere in the universe that has 6 protons in its nucleus we would classify that as carbon. Now, carbon as well has 6 electrons found around the nucleus of that atom. 2 of those electrons are found in that first orbit, and then the remaining 4 electrons are found in the second orbit around that nucleus. And then you'll note that carbon does have room for 4 more electrons within that second orbit. So why is this significant? Well, this is what carbon can use to create 4 bonds. So it has those 4 electrons in that second orbit. It has room for 4 more. And ultimately, all atoms want to gain a full outer shell of electrons. For carbon, this is a total of 8 electrons in that second orbit. Now, atoms can get a full outer shell through a couple of different ways. So they can lose or gain electrons, and this is how ionic compounds are formed. Or they can gain electrons by sharing electrons with other atoms, and this is how a covalent bond is formed. So carbon doesn't lose or gain electrons in this case, but it will create covalent bonds by sharing electrons in its outer shell with other atoms. So carbon has 4 electrons in that outer shell and can thus form 4 covalent bonds. So in the diagram, the blue dots represent the electrons that carbon has in its outer shell. So it has the 1, 2, 3, 4 electrons. And then each hydrogen atom here has 1 electron that it can share, which is represented by the red dot. So now, by creating a covalent bond with 4 different hydrogen atoms now, carbon can get the full 8 electrons that it needs in its outer shell to be happy. And hydrogen can get 2 electrons in that first outer shell, and then hydrogen is happy as well. Everybody's happy. So there are different types of covalent bonds that can be formed. So there is the single covalent bond, or just a single bond. This is where 1 pair of shared electrons is shared between 2 atoms. If you have a double bond, this is where you have 2 pairs of shared electrons between 2 atoms. And just note that a double bond is represented by 2 lines between the atoms. A single bond is just represented by 1 line between the 2 atoms. And we use that line just to represent that there are electrons being shared between those 2 atoms. Now, you can have a triple bond where you get 3 pairs of shared electrons between the 2 atoms, and then this is represented by 3 lines between those atoms. So the bonds that are created now can be with other carbon atoms or with other atoms such as hydrogen, oxygen, nitrogen, phosphorus, or sulfur. And these are the common atoms that we'll find making up these organic compounds, with carbon as the central atom, or the atom that everything else is bound to. So there are 4 main classes of carbon compounds, or 4 main categories of carbon compounds that can be found. There are the carbohydrates, such as glucose and fructose, and these are composed of carbon, oxygen, and hydrogen. So you'll only find those 3 elements in carbohydrates. And hydrogen and oxygen will usually be found in a ratio of 2 to 1. Another category of carbon compounds that we can have are the lipids, which are insoluble in water. And lipids will have a long chain of carbons attached, anywhere between 12 to 18 carbons attached in a row. Another category are proteins, which are composed of molecules referred to as amino acids. Amino acids are the individual molecules that go together to make up a protein. The general structure of an amino acid is found in the diagram here. But we'll go through what those different parts are later on in the video. These amino acids, or these proteins, will contain carbon, hydrogen, oxygen, will contain nitrogen, and two of them will contain sulfur. Two of those amino acids will contain sulfur, but every amino acid will contain carbon, hydrogen, oxygen, and nitrogen. Nucleic acids, which will be classified as DNA and RNA, will contain carbon, hydrogen, oxygen, nitrogen, but will also contain the atom phosphorus as well, and will not contain sulfur. So one of the skills that you will be required to do is to be able to draw a number of, draw molecular diagrams of the number of different molecules, which include things like glucose, ribose, saturated fatty acid, and a generalized amino acid. So when drawing glucose that has the molecular formula C6H12O6, this is a six-membered ring with one side, and we'll make a drawing to see what that looks like. So when we say a six-membered ring with one side chain, based on the molecular formula, glucose will have that six-membered ring of oxygen. I will start my drawings with oxygen that has a single covalent bond to carbon, which is covalently bonded to another carbon. Carbons. So we have our six-membered ring. So if you count up the oxygen with the carbons, you get a total of 6 elements in there. And then the side chain comes off of the top carbon, and this top carbon, and then the side chain is just the remaining carbon and hydrogen and oxygen molecules. So it's a CH2OH as the side chain. So you have one carbon atom that forms the side chain, and then the carbon atoms can also be numbered, but we'll number those in a second. We'll finish our structure first. So carbon, or glucose, has what's called hydroxyl groups, which are these OH groups. And on carbon atoms 1 and 2, they can point, they do point down. Point them down. On carbon 3, they will point up. On carbon 4, they will point down. Now, remember that carbon can form 4 bonds, and right now we have carbon only bonded to 3 things. So to create that 4th bond, we simply fill in hydrogen as that remaining bond. So you'll see in the diagram that each carbon is now bonded to 4 separate things, and we can also label our carbon. So the carbon to the right of oxygen would be labeled as carbon 1, and then we go around clockwise labeling our carbons. Carbon 2, 3, 4, 5, and then carbon 6 makes up the side chain in glucose. Please keep in mind that how you draw your covalent bonds between elements is important. Remember, the lines show sharing of electrons between elements. So when drawing a covalent bond between carbon and a hydroxyl group, for important, it is important to show the line between the carbon and the oxygen. This is correct. if the line is drawn in between carbon and the hydroxyl group, or shown with the hydrogen, this is actually incorrect because the covalent bond is not between carbon and hydrogen, but between carbon and oxygen. In terms of the molecular drawings for ribose, ribose is going to be another ring structure composed of 4 carbon atoms and 1 oxygen atom. So ribose will have the following structure to it. So we'll have an oxygen, carbon, and carbon. So it will be a 5-membered ring, and it will have that 1 side chain. We'll have CH2OH, and then just note that I've drawn my my covalent bond between the carbon and the side group, between the carbons. And then we'll have hydroxyl groups on each of the carbons. And then the direction that the hydroxyl group is important. So in this case, on carbons 2 and 3, the hydroxyl groups will point down. And then we'll have hydrogens that fill in the rest of those covalent bonds. And then, as with glucose, we can also number our carbons. So the carbon to the right of oxygen gets labeled as number 1, and then we just number the rest. So the orientation of the hydroxyl groups relative to the carbon, relative to the hydrogens, is important in ribose and glucose. And then keep in mind that only the ring form of ribose is needed. So there is a straight chain form of it, but we only need to be able to draw the ring forms of ribose and glucose. And then glucose has 2 forms to it. There is an alpha glucose and a beta glucose. And the difference is at carbon number 1, the orientation of the hydroxyl group. So if the hydroxyl group is pointing one way, it's an alpha glucose, and then if it's pointing a different direction on that first carbon, it would be beta glucose. But as long as you draw the hydroxyl group pointing up or pointing down on carbon 1, it doesn't matter. The rest of the carbons do matter, the direction of the hydroxyl group and which way they are pointing. For the saturated fatty acids, this one can be a little bit time-consuming. But when drawing a saturated fatty acid, just keep in mind that the carbon atoms will form an unbranched chain, and it will be single bonds in the saturated fatty acid. In a separate video, we'll go through the difference between a saturated fatty acid and an unsaturated fatty acid, but right now it's just single bonds between each of the carbons in that chain. And the number of carbon atoms can be anywhere between 14 to 20. So if you draw 14, it's fine. At the end of, at one end is going to be a functional group referred to as a carboxyl group. That carboxyl group is defined as a carbon double bonded to an oxygen, and then that same carbon is bonded to a hydroxyl group. And then the fourth bond will be that carbon attached to that chain of carbons. And then at the other end is a carbon atom bonded to 3 hydrogens. So you'll have a carbon that is bonded to 3 hydrogens, and then this carbon is going to be attached to the chain of carbons. And then every other carbon atom within that chain is going to be bonded to two hydrogen atoms. So what would a drawing of this look like? So at one end, you would have your hydroxyl group, your carboxyl group, sorry. And then you would have a chain of carbons. So I have 12 carbons in my chain. The very last carbon, I would draw 3 hydrogens attached to that carbon. And then every other carbon would be bonded to 2 hydrogen atoms. And we just fill in the hydrogens for the rest of those carbons. What I mean when this is a little bit tedious. But we don't need to remember direction of hydroxyl groups in saturated fatty acids. So here we have our saturated fatty acid. And it is only expected that one saturated fatty acid be drawn, and the specific name is not necessary. So you'll just be asked to draw a saturated fatty acid, and then you would make a drawing similar to what we've just done. In the last of the diagrams, you'd be expected to be able to draw a general amino acid structure. So an amino acid is basically a carbon atom that's bonded to 4 different things. And it's very specific what those 4 things are. So you have an amine group, which is just nitrogen, covalently bonded to 2 hydrogen atoms, and then that nitrogen atom is bonded to that central carbon atom. You have a carboxyl group, which is the same carboxyl group that we saw in the saturated fatty acid, and then you have the hydrogen atom that's also bonded to that. And then the 4th thing that's bonded to that carbon in an amino acid is what's referred to as an R group, which is the variable part of the amino acid. It's what determines which amino acid we are dealing with. So there are 20 common amino acids, so there are 20 different R groups. We are not expected to know those R groups. They do not have to be memorized. So we'll just include an R to represent that variable portion of the structure. So when we draw an amino acid, we can draw it as our central carbon that is attached to that hydrogen atom. Below it is going to be the R group, which we just represent with an R. To the right, we can draw our carboxyl group. And then to the left, we can draw our amine group. And so carboxyl groups are also acidic. The carboxyl group is what contributes to the acid part of the name. So we have an amine group, which is also at this point called an amino group. So we have an amino acid. So you'll also be expected to be able to identify molecules as a carbohydrate, lipid, or amino acid. And there are a few tips for being able to identify each of these compounds. So keep in mind that carbohydrates do not contain nitrogen. So if there's no nitrogen present, it's going to be a carbohydrate. And carbohydrates contain hydrogen and oxygen in that 2-to-1 ratio. So if you look at the molecular formula for glucose, you'll see that hydrogen and oxygen exist in that 2-to-1 ratio. And then lipids do contain relatively less oxygen than carbohydrates. So lipids do not contain hydrogen and oxygen in that 2-to-1 ratio. So if you look at this chemical formula for a fatty acid, you know, the hydrogen to oxygen ratio is 14 to 1. So nowhere close to that 2 to 1 ratio. Now, you'll also should be able to recognize from molecular diagrams that other types of molecules are classified as lipids. Drawings of these are not expected, but you should be able to categorize them as lipids. So these molecules include triglycerides, which are basically three fatty acid molecules attached to a glycerol molecule. You can recognize the triglyceride by the long chains of carbons, and there's 3 of them. There is also the phospholipid structure, so that same phospholipid that goes into making up that cell membrane, you should be able to recognize that as a lipid. So again, long carbon chains found within the molecule, and then steroids are also considered lipids as well. So for example, cholesterol, which was discussed in making up cell membrane structure, is a lipid as well. And so, you know, you can recognize the steroid by the general ring structures that they have attached together. So you should also be able to recognize proteins or parts of polypeptides. Recognize that these would be classified as amino acids, or you know, they would be made up of amino acids. So proteins, and a three-dimensional diagram is shown of a rather simple protein, but even the rather simple ones are complex. And if you string a bunch of amino acids together, you create what is called a polypeptide. And so polypeptides contain a special type of bond referred to as a peptide bond, which we'll cover in more detail when we talk about proteins. So ultimately, a big part of molecular biology is dealing with metabolism. And so metabolism is basically the web of all enzyme-catalyzed reactions in a cell or organism. So all living organisms perform many different chemical reactions. And these reactions are all catalyzed by enzymes. So what we mean by this, by all these chemical reactions, is that one type of molecule is going to be changed into another molecule in a series of small steps. So you start with one reactant and make a slight change to it in one step, and the next step another change is made to it, and a series of small changes are made until you've now created this new molecule. So in these series of reactions, they can occur as a chain of reactions or as a cycle. So what we mean as a chain is that you start with molecule A, this gets changed to B, C, and then you end up with product D. This occurs in a linear fashion. And when we talk about these reactions occurring in a cycle, this is where you can have product D turned into E, which gets turned into molecule F, which molecule F then gets turned into G. But G is then fed back into the cycle, so G can be used for other types of reactions or used as a product, but then G is used along with D to create that first part of that series of reactions. So you end up with a cycle of reactions occurring. All of these reactions can basically be divided into 2 parts. So reactions can be considered as anabolic reactions or catabolic reactions. So in anabolism, these are reactions that will create larger molecules from smaller molecules. So you take a bunch of small molecules and you build something bigger at the end. To do this, it will require an input of energy in the form of ATP. And so examples of this in a cell would include things like protein synthesis. When DNA is made, DNA is made up of a bunch of small molecules being joined together. That process of photosynthesis, where you take carbon dioxide and water to create glucose. Is an example of an anabolic reaction, or the synthesis of complex carbohydrates, such as cellulose, for example. In catabolism, this is where larger molecules now get broken down into smaller molecules. In general, they will release energy, which sometimes can be captured in the form of ATP, or which can be used to drive the creation of ATP. So examples include like the digestion of food, cellular respiration, where glucose is taken, so you take that big molecule, and then you break it down into that smaller molecule now of carbon dioxide and water. Or, for example, decomposers breaking down complex carbon compounds into smaller molecules. So that is the end for this video of 2.1.